At the end of the lecture, the student will be able to describe the functions of different respiratory centers, the chemical control of respiration, and the different factors affecting the rate of respiration. The central nervous system has the ability to control the rate of alveolar ventilation exactly according to the demands of the body. And this function is provided by collection of different neurons which are located mainly in the pons and the medulla oblongata. These neurons are classified according to their function as dorsal and ventral respiratory group of neurons which are located within the medulla oblongata, and within the pons there are two other centers which are pneumotaxic and apneustic center. Beside these centers, there is also the chemical control of respiration which is provided by chemoreceptors. These chemoreceptors are centrally placed within the medulla oblongata, and there are also the peripheral chemoreceptors which are located in the aortic and carotid bodies. The spinal cord is also associated with the regulation of voluntary breathing, and it is controlled by the cerebral cortex. So within the medulla there are the dorsal and ventral respiratory group of neurons. The dorsal respiratory group of neurons is associated with the inspiratory ramp signals. It means it has the function within the inspiration. And the ventral respiratory group, it is the expiratory center. It is mainly active in forceful breathing or active breathing. The pontine centers are pneumotaxic center and the apneustic center. So these are the areas within the brain: pneumotaxic center and the apneustic center within the pons and the medulla oblongata. The dorsal respiratory group of neurons are located in nucleus tractus solitarius. Additional neurons are also located in the reticular substance of the medulla. It provides the basic rhythm of respiration. It means it generates the inspiratory ramp signals. So this is the dorsal group of neurons located dorsally in the medulla oblongata, and it is here that the inspiratory signals are initiated, and initially the signals are weak and steadily and gradually these signals are increased in their strength. So the name is inspiratory ramp signals. They increase in a ramp like manner. The nervous signals begin weakly and transmitted to the inspiratory muscles, mainly the diaphragm. It increases in a ramp manner for about 2 seconds. Then it ceases for next 3 seconds, allowing the diaphragm to relax and causes the elastic recoil of lungs and chest wall to expire outside the lungs. So inspiration, the duration is about 2 seconds, and then there is the expiration which is of 3 seconds. There is the cycle of 2 seconds of inspiration and 3 seconds of expiration. The significance of inspiratory ramp signal is that the lungs fill rapidly but smoothly. It regulates the rate of respiration by seizing the ramp signals. Rate of respiration is increased because if the inspiratory ramp signals are stopped, then ultimately the expiration will be more rapidly, and the whole cycle of inspiration and expiration will ceases. So this is the whole cycle of breathing, normal breathing. This is the dorsal respiratory group of neurons. It provides the inspiratory ramp signals. the muscles of inspiration, mainly the diaphragm, contracts, inspiration occurs, then the dorsal respiratory group of neurons are inhibited to cause the expiration by the elastic recoil of lungs. The ventral respiratory group of neuron is active during active breathing, during forceful breathing, and it causes both expiration and inspiration. The nucleus is found in nucleus ambiguous rostrally and retroambiguous caudally. In forceful expiration, not only the elastic recoil of lung is needed, but also the internal intercostals and abdominal muscles are required to contract for forceful breathing. So this is the ventral respiratory group of neurons. It is active during forceful breathing and causes expiration and inspiration, located ventrally in the medulla. So you can see here, this is the ventral respiratory group of neurons. It causes the activation of the internal intercostal muscles and the abdominal muscles to cause the expiration. Whereas this is the dorsal respiratory group of neurons. It mainly causes the activation of the diaphragm and external intercostal muscles, which also helps during the inspiratory process. Two other centers within the pontine area are the mnemotaxic center and apneustic center. The mnemotaxic center sends inhibitory signals to the dorsal respiratory group of neurons. It means it causes the control of switching of the inspiratory ramp signals. And another is the apneustic center. Apneustic center causes the inactivation of signals which are coming from the mnemotaxic center. So it prevents the switching off of the inspiratory ramp signals. This is the mnemotaxic center located in the nucleus parabrachialis of upper pons. Transmits signals to the dorsal respiratory group to switch off inspiratory ramp signals. In this way, it limits inspiration and increases the rate of breathing. Strong signals can increase the rate of breathing up to 30 to 40 breaths per minute, and weak signals can cause the respiratory rate of about 3 to 5 breaths per minute. This is another diagram showing the role of the dorsal respiratory group of neurons supplying the diaphragm to cause inspiration, and the ventral respiratory group of neurons supplying the muscles of expiration, which are the internal intercostal muscles and abdominal muscles. Apneustic center is located in the lower pons. It inhibits the signals from the mnemotaxic center and prevents switching off of dorsal respiratory group. Hence, it prolongs the inspiration with shortened expiration. Occurs in severe types of brain damage. Now, the chemical control of respiration depends upon the levels of carbon dioxide and hydrogen within the blood. Under normal conditions, 75 to 85% of respiratory drive is due to the stimulation of central chemoreceptors by carbon dioxide. So carbon dioxide provides the potent regulator of chemical control of respiration. The central chemoreceptors are directly stimulated only by hydrogen ions. But due to the blood-brain barrier, the hydrogen ions cannot cross the barrier while the carbon dioxide can cross it. So carbon dioxide stimulates the respiratory center more than the hydrogen ions. This is the chemosensitive area, which is located beneath the ventral surface of the medulla. This area is highly sensitive to changes in partial pressure of carbon dioxide and hydrogen ion concentration and excites the respiratory centers. The hydrogen ions are the most potent stimulator of the chemosensitive area. But the hydrogen ions, if they are increased in the blood, they cannot cross the blood-brain barrier. Ultimately, carbon dioxide, if its levels are increased within the blood, they can cross the blood-brain barrier, but they combine with the water of the tissues of the brain to form the carbonic acid, which then dissociates to form the bicarbonate ions and hydrogen ions. And then the hydrogen ions, they act on the chemosensitive area of the brain. So this is the chemical control of respiration within the blood. If the partial pressure of carbon dioxide is increased, it enters the blood-brain barrier very conveniently and it binds with the central chemoreceptors, which lie just beneath the ventral respiratory group of neurons, and here it activates the respiratory centers to cause the increased ventilation. Another diagram. This is the blood-brain barrier here. Partial pressure of carbon dioxide increases. The carbon dioxide combines with water to form carbonic acid, which dissociates into hydrogen ion and bicarbonate ions. The hydrogen ion then stimulates the central chemoreceptors. The signals are sent to the respiratory control centers to increase the ventilation. This is the same diagram, making you more clearer about the chemical control of respiration. If the hydrogen concentration is increased in the blood, it can't enter the blood-brain barrier. So you can see here hydrogen ions cannot enter the blood-brain barrier, and that's why carbon dioxide ultimately stimulates the central chemoreceptors within the chemosensitive area, which is located in the medulla oblongata. The peripheral chemoreceptors are stimulated by the oxygen concentration. The central chemoreceptors are stimulated by carbon dioxide and hydrogen ions, whereas the peripheral chemoreceptors are stimulated more potently by the concentration of oxygen within the blood. The carbon dioxide and hydrogen also can stimulate the peripheral chemoreceptors. These peripheral chemoreceptors are located within the carotid body, which is present at the bifurcation of the carotid artery, and the aortic bodies, which are present at the arc of the aorta. From the arc of the aorta, the sensations are carried. If there is decreased oxygen, the sensations are carried by the vagus nerve to the respiratory center. And from the carotid body, the sensations are carried through the hurrying nerves then the glossopharyngeal nerve, and ultimately into the respiratory centers to increase the pulmonary ventilation. The carotid and aortic bodies are sensitive to decrease in partial pressure of oxygen, an increase in partial pressure of carbon dioxide or hydrogen ion concentration. They are maximally stimulated when the partial pressure of oxygen decreases below 50 to 60 millimeters of mercury. They detect changes in dissolved oxygen but not in the oxygen that is bound to hemoglobin. Beside the central and peripheral chemoreceptors and the 4 respiratory centers, there are other reflexes which regulate the respiration, in which there is the Hering-Brewer reflex, baroreceptor reflex, cough and sneezing reflex, and lung J receptors. More important is the Hering-Brewer inflation reflex, which is stimulated when there is excess inspiration. The lungs become overstretched, and it is activated when the tidal volume increases up to 3 times normal, that is 1.5 liters per breath. The normal tidal volume is 500 milliliter. If it is increased up to 1500 milliliters per breath, then this reflex is initiated. The stretch receptors located in the muscular portion of the walls of the bronchi and bronchioles are stretched and transmit inhibitory signals to the dorsal respiratory group of neurons and inhibit further inspiration. It is a protective mechanism and prevent excess lung inflation, and it is more important in neonates. Others include the baroreceptor reflex, which are associated with increase in the arterial blood pressure. There are also the J receptors, which are located in the alveoli, and these receptors are stimulated due to the edema or the emboli, causing the pressure on the capillaries of the alveoli. These receptors are responsible for the sensation of air hunger, dyspnea, or shortness of breath. In case of any allergen or irritant substance entering the airway, there is stimulation of the receptors in the upper airways. The afferent signals via vagus or the trigeminal or olfactory, they send to the respiratory centers to cause the deep inspiration followed by forced expiration against the closed glottis to cause the cough or sneezing reflex. This is the control of overall respiratory center activity. The higher centers of the brain, they are in continuous association with the different types of receptors within the heart, lungs, muscles, and also different brain areas regulate the respiratory rate and pulmonary ventilation. The respiration is also regulated during exercise and hyperventilation. It is a learned response, and the neural input from the motor cortex also supplies the muscles which increase their activity during the exercise, also increase the respiratory rate. The proprioceptors in moving muscles and joints also stimulate the respiratory centers. If there is an increase in the body temperature, it also stimulates the respiratory centers. The catecholamines stimulates the respiratory centers. The pH changes due to lactic acid during muscle exercise is another stimulus for the increased pulmonary ventilation.